So in last video, we talked about the monomer of protein, right? So the monomer of protein is a molecule which we call amino acid. So if when you see this amino acid, you can kind of get a sense of what is it actually made of, right? It is a biomolecule, so it should be carbon-based. And you're correct. Amino acid is carbon-based. It is actually made of a carbon being the central atom and is attached to 4 different groups to that particular central atom. So the 4 groups that's attached to the carbons are what we call the amino group. So amino group, hence amino acid gets its name, amino group. Next, it is also an acid, and we know that one of the particular functional group that contributes to the acidity of a molecule, for instance, the fatty acid, right? The fatty acid has a carboxyl group. So an amino acid also has a carboxyl group. So carboxyl group. So now we have 2 groups, right? Amino group and carboxyl group. Right on top of the carbon is 1 hydrogen atom, just an H. The last one that's really confusing, but it's also contributing to the diverse structure of protein and amino acids, is this what we call the R groups. Why do we call it an R groups? It's not really a functional group per se, but rather it's composed of 20 different types of groups. This R groups has 20 of them. And depends on what type of R group you have for that particular amino acids is going to give different properties for that particular amino acids. Some of them are hydrophilic, so hydro means water, philic means like. Some of them are hydrophobic. Hydro means water, phobic means don't like, fear. Some of them has charge. They are positively charged or negatively charged. Some of them are polar. It means they are slightly charged. And some of them are just neutral. And some of them have ring shapes in it. Just like the carbohydrates, the glucose, it has some sort of carbon rings in there. So it is really diverse. It depends on what kind of amino acids you have. You're going to form different bonds between the amino acids. They're going to form different attractions between these R groups, right? You know that charge attracts charge. Hydrophilic is going to group with hydrophilics, especially water. And hydrophobic is going to run away from water. So as you can see, this R group is very diverse. The specific structure of the R group is not the focus of this class, but it gives you a perspective on how important this R groups is and how they can contribute to the 3D shape of a protein. The focus that we wanted to do is the specific structure of amino acids. So let's redraw the picture. So central carbon, right on top is hydrogen, right below is the R group. To the left is the amino group. So amino group has 1 nitrogen and 2 hydrogen attached to the nitrogen. The carboxyl group has 1 carbon and a double bond oxygen and a hydroxyl group. This is a carboxyl group, carboxyl, and this is an amino group, So now I'm just going to redraw amino acids and put it side by side with each other. So now if you look closer, I'm putting 2 amino acids side by side. What do you realize? You realize that there is a hydroxyl group in the carboxyl and a hydrogen in the amino, right? When we put them side by side, they can actually become water. So right now I'm going to remove a hydroxyl. So I'm going to take this hydroxyl, and take this hydrogen away. Take this hydrogen away. So now they form bond. Hydrogen stick to a hydroxyl. It becomes a water molecule, right? A water molecule, H2O. The removal of this water molecule is going to create a bond between the carbon and the nitrogen, between the carboxyl and the amino group of 2 different amino acids. So now what I just did is a removal of water molecule, right? to form a new thing. Now there are two amino acids connected to each other. 2, the prefix for 2 in biology is di. So I just make a dipeptide. Peptide is protein. So a dipeptide, DTE, dipeptide. And this bond, we mentioned it before in the previous video, this is a special covalent bond between 2 amino acids to form a polymer. So this is what we call a peptide bond. And this whole process is called dehydration synthesis. Dehydration synthesis. And if you keep repeating this process, eventually you're gonna make a polypeptide from many amino acids through the removal of water, and that process is called dehydration synthesis.